Hello everybody. In my last lecture, I talked about the visual pathway, how the visual signal is taken from the retina of the eye to the primary visual cortex or V1, right? So today, first I'll talk a little bit more about the retina, retinal ganglion cells, and then the pathway, and then mostly I'll talk about the primary visual cortex. Okay. So from my last lecture, you must remember that there are three layers of the cells in the retina: the photoreceptors, rods and cones, right? That is one layer. And then you have the bipolar cell layer, and then innermost layer is the ganglion cell layer, right? And the ganglion cells receive the signal from the bipolar cells. These are the photoreceptors, okay? Ganglion cells. And the axons of ganglion cells form the optic nerve, okay? Cranial nerve number 2. Okay. However, I did not show you what kind of connectivity could be present between these cells. Although I showed you the horizontal cells and amacrine cells, those are the cells, they modulate the signal transmission in the retina. Here in this slide you can see that how the photoreceptors converts the signal, visual signal, to the ganglion cells. So these are the photoreceptors, they have shown the cones, and these are the ganglion cells, okay? So if you see here how the convergence can occur on the ganglion cell. Here you can see the spatial summation, and here you can see the acuity. First, let's see how the convergence can occur on the ganglion cells. The yellow area is showing the light or stimulus, visual stimulus, the incoming light. You know that light goes this way, right, to stimulate the photoreceptors, okay? So here this light, incoming light, stimulates all these photoreceptors, okay? And all these photoreceptors are converged on this retinal ganglion cell, so on this single ganglion cell. So input from these 4 cones or photoreceptors is going to this ganglion cell, right? So that will excite the ganglion cell, and you see the action potentials here, right? That will be taken to the brain. Here the stimulus is narrower than this one, right? So in this case, if the stimulus is narrow, that would only, this is like almost half of this stimulus, so it will stimulate only these 2. That means fewer cones. And from these 2, in this case, from these 2, the convergence would occur on this ganglion cell, right? So that will also activate the ganglion cell, but the firing will be less than or weaker than this one. So more convergence, more activation of the ganglion cells. Make sense? That means the brain will get stronger signal. Here, in this case, this could be another, way the ganglion cell is activated. You see here, this is the stimulus, which is really narrow compared to other previous two, right? So this area of stimulus will activate only a single cone. Here there is no convergence, right? So only one cone is giving signal to one ganglion cell. Okay, so in this case the ganglion cell will fire or produce action potential, but much weaker response. Okay, so more convergence, more activation of the ganglion cell, that is the strength of the stimulus. Make sense? That could be one type of activation of the ganglion cells. Now you see spatial summation, the location. So this is again the incoming light or stimulus, and this one is activating this cone. Okay, so the ganglion cell will be stimulated, but weakly stimulated, right? Now if you present the stimulus in a way that it activates these two cones, right? So the convergence here is more, and that will activate the ganglion cell more. So you see, if you shift the location of the stimulus, which area of the retina or which photoreceptors are being activated, right, that can be detected by the response of the ganglion cell here. In this case, in all these three, you see that the connectivity is same, right? That means four cones are being converged on the same ganglion cell. Here in the one before, you saw this is more convergence, intermediate convergence, or moderate convergence, and no convergence here, right? So the response depends on the convergence, how many neurons, how many photoreceptors are giving signal to that ganglion cell. That is one. Here you see, although the same connectivity, same situation, same amount of convergence, however the ganglion cell is being activated by the stimulus, and the number of photoreceptors are being activated. Makes sense? So that is spatial summation that will tell where the stimulus is. And now here the acuity depends on the input to the ganglion cell. Here you see high convergence, that means four photoreceptors cones are giving signal to this ganglion cell in both cases. However, if you present the stimulus that activates these two cones, the response of the ganglion cell will be like this. And if you move the stimulus, however, still that stimulus is activating two photoreceptors, same number of photoreceptors, but different photoreceptors that will activate same way, because two cones are giving signal to this ganglion cell. Make sense? So this is high convergence situation. Now no convergence here. You see that one photoreceptor is giving signal to one ganglion cell. So although here the stimulus is same in these two cases, right? The stimulus in this case will activate two separate ganglion cells. Two cones will activate two separate ganglion cells, and the signal will go to the brain separately. In this case, from these two photoreceptors, the signal is converged here, so the brain will get single signal from this single ganglion cell, right? So brain will not be able to determine the two separate objects in this case because they are being converged here. So brain cannot get the signal separately, brain cannot process the signal separately. So brain will not be able to tell the two separate points. In this case, since the brain is receiving the input separately, the brain will be able to tell the difference, will be able to discriminate two different objects, although they are located, in the same space. Okay. So this is more in the fovea or central part of the retina, where the cones have less convergence. That means the brain receives signal from separate cones separately, and that gives the high acuity, the ability to discriminate the small things more clearly. Makes sense? In the outer part, as you move to the outer part, more convergence and less acuity. Okay. So this is actually showing that how the connectivity in the retina can activate the ganglion cells differently. Okay. Here you can see that how the retinal ganglion cells can detect the edges. You must remember that I explained earlier, the retinal ganglion cells have on center of surround, one type of retinal ganglion cells. And here you can see 4 on center of surround retinal ganglion cell. And now if I present a light that will fall to the center, that means on center, that will excite this ganglion cell, right? So you will see the firing action potential. Okay, so this is the condition when I am putting the stimulus just on the center. So this is on and this is off surround, right? Now if I present the stimulus only here to the off surround area, then what will happen? No response, because this area will be inhibited by the light or stimulus. Make sense? So this is condition 1, this is condition 2. Now if I present the stimulus or light in a way that will fall on the center as well as on the outer of area of surround. Then what will happen? They will cancel each other. On will excite, but also since the stimulus is on the surround, that will inhibit. So excitation and inhibition together, right? So that will do what? Will neutralize or cancel each other. So you will see only few spontaneous firing. Okay. So this is strong excitation, this is strong inhibition, this is neutral, I can say. Now, if I don't put any stimulus, no light, so both on and off in darkness, both on, dark will stimulate the off surround but inhibit the on center. So it's just opposite of this, right? So this is also neutral. They will cancel each other. Makes sense? So this is in light condition, this is in dark condition. In light condition, right, the on will be activated, off surround will be inhibited. They will cancel each other. In dark condition, the on will be inhibited, but off surround will be activated. They will cancel each other. So you see that the ganglion cell could be activated in all different ways or inhibited. So now let's see here. This is the stimulus. Here you have a dark part and a light part. Okay, this is dark, this is light. Now these are the ganglion cells. 4 different ganglion cells, right? Now let's see this condition. Here you have light, so on center will be activated, off surround will be inhibited, so they will cancel each other. So you'll see only few action potential, right, like this. Now let's see this one. Here you have dark stimulus, so the off surround will be activated, on center will be inhibited. Makes sense? So they will cancel each other. So similar response in light and dark. So by looking at these two, the brain will not be able to tell the difference, because brain cannot see what is going on outside. Brain just only counts the number of action potential, right? So in this case, the brain will not get any information. However, those ganglion cells are in this area where the edge is. Now you see they will be activated differently. In these two cases, I can say that the surround, off surround, in this case the off surround is activated here by the dark, and in this case also off surround will be activated by the dark, right? And here the off surround will be inhibited by light, here off surround will be inhibited by the light. So no difference. Part of it is being excited and part is inhibited. So they will cancel each other. The difference is this one and this one, the on center. So in this case, the on center will be fully activated by the light. And in this case, the on center will be inhibited strongly by the dark, right? And that's why you see in this case the response is high, strong, and in this case the response is low or very weak. Okay, no response. So when the brain will receive these two, the brain will be able to detect the difference, right, by counting the action potential, and that will help the brain to determine the age. So this is how the ganglion cells can help the brain to detect the ages. Make sense? And stripes. Okay, so you now know how the photoreceptors can, activate the ganglion cells by convergence, by spatial summation, and by the acuity. Okay. Now let's talk about the visual pathway. You already know the visual pathway. We have talked about that. So from the ganglion cells of the retina, the axons converge to form the optic nerve, and through the optic disc, the optic nerve exists and become myelinated, right? And optic nerve join to form the optic chiasm, and then the optic tract. Now the optic tract, you must remember that, let's draw here. So this is the lateral, these two are lateral geniculate nucleus in the thalamus, right? And two eyes. Okay, so we know that crossover here, and also ipsilateral, right? Fibers go like this. Okay, so this is optic nerve, optic chiasm, optic tract, right? You already know. And the optic tract carries the signal to the lateral geniculate nucleus. And you also remember that lateral geniculate nucleus has 6 layers. Okay, 1, 2, 3, 4, 5. There should be one more. Same this way, 6 layers. Okay, and layer 1, 4, and 6 get signal from contralateral, that means opposite eye, and 2, 3, and 5 receive signal from the ipsilateral. Okay, so the input from 2 eyes are kept separated in the lateral geniculate nucleus, right? And the layer 1 and 2, these 2 layers are called magnocellular. So these are magnocellular layers. And layer 3, 4, 5, 6 is here. Layer 3, 4, 5, 6 are called parvocellular. So these are parvocellular layers. Okay, parvocellular. And each of these 6 layers has a kinocellular layer. So this is kino. So 6 kinocellular layers. Okay, so kinocellular. So those are the layers in the lateral geniculate nucleus. Okay, and you must remember 3 major type of cells, ganglion cells, are the midgate, right, paracel, and bistriate. So they send signal to these layers. And also you must remember from the lateral geniculate nucleus, optic radiation or geniculoplasty calcarine tracts take the signal to the primary visual cortex. So this is the primary visual cortex. Okay. Primary visual cortices receive the signal from the lateral geniculate nucleus through optic radiation or geniculocalcarine tracts or fibers. And these fibers go through different pathways. This is the parietal or superior through the parietal lobe, and this is the temporal inferior fibers. Okay, so that's how the signal is taken to the primary visual cortex or V1. Okay, and V1 is also known as Brodmann area number 17. You know that different functional areas in the cerebral cortex are given different numbers based on the functions. So the person Brodmann, who investigated the functions of different parts of the cerebral cortex, and different areas were given different numbers, and area number 17 is the primary visual cortex, which is also called striate cortex because of the striation present in the V1. Okay, so that's how the visual signal is taken to the V1, that means the sight or image. However, you also know that from the lateral geniculate nucleus, some fibers go to the superior colliculus and also to the optic superior pretectal area, pretectal nucleus. Okay, so superior colliculus and pretectal nucleus receive the fibers from the lateral geniculate nucleus, and superior colliculus send signal to the nuclei of the cranial nerves. Those cranial nerves control the extrinsic eye muscles. So this is the oculomotor nucleus, trochlear nucleus, and abducens. So oculomotor, trochlear, abducens. Okay, and they are present in the brainstem, and these two are located in the midbrain, and the abducens nucleus is located in the pons. Okay, so this one in the pons, and these two are in the midbrain. Anyway, so from these three cranial nerve nuclei, the signal go to the extraocular muscles. They regulate the movement of the eye. Makes sense? Now, this is kind of repetition I already mentioned. From the pretectal nucleus, that means from here, the signal goes to the Edinger-Westphal nucleus. Here some crossover take place, right? So that's why if I stimulate one eye, the other eye responds to same way. Okay, so crossover. So these are Edinger-Westphal nucleus. Parasympathetic centers. And from the Edinger-Westphal, the parasympathetic nerve fibers enter into the eyeball and innervate the ciliary body and the iris to regulate the shape of the lens for accommodation and size of the pupil. Okay, so ciliary body or ciliaris muscle and iris. Okay, so when this nucleus, Edinger-Westphal, is stimulated, parasympathetic innervation activates the ciliary body that will make the lens more round. Okay, and constrict the pupil. That means make the pupil small. Okay, so the lens will be more round, more accommodation for near object. That means the near object will be better, or more clearly seen, and constriction of the pupil will also regulate the light. Okay, so that is how the visual pathway, activate different structures. Now, if you see the cranial nerve nuclei here that I already explained, right? So this is the midbrain, this is also midbrain, right? And this is the oculomotor nucleus, this is the trochlear nucleus. Both are in the midbrain. However, this is the pons where you have the abducens nucleus. Okay, and from these nuclei, the cranial nerves arise and go to the extrinsic eye muscles. And you already know that oculomotor nerve activates the 4 muscles. This one, this one, this one, and this one, right? So superior rectus, inferior oblique, right? And also the lateral rectus and inferior rectus. And the trochlear nerve, this one, innervates the superior oblique. Okay, and the abducens, this one, activates the lateral rectus. Here, I already mentioned that there are 6 layers in each lateral geniculate nucleus, right? And the layers are parvocellular, magnocellular, and kinocellular, shown by 3 different colors. So you can see here, magnocellular, I mentioned layer 1 and 2, these 2, right? And you remember I mentioned parvocellular layer 3, 4, 5, 6. So 3, 4, 5, and 6. And kinocellular in all 6. So you see the green color is showing the magnocellular layers 1 and 2. The pink color is showing the parvocellular 3, 4, 5, 6. And the cyan color, light blue, is showing the kinocellular layers. Makes sense? And you also see that layer 1, 4, 6 receive input from the contralateral, and layer 2, 3, 5 receive signal from the ipsilateral eye. Okay. And also, we already know 3 types of ganglion cells send signal to these layers: midgate, paracel, and bistratified. Here you see this is the real slice picture of a lateral geniculate nucleus. So you can see how the layers are separated by connective tissue membranes, and the input from 2 different eyes are kept separated, that I mentioned, in these layers. Now, how a smaller number of lateral geniculate nucleus neurons provide information to many V1 neurons, because the number of V1 neurons is much higher than the number of neurons in the lateral geniculate nucleus. So this is a hypothesis that explains how the same neuron of the lateral geniculate nucleus can participate in different stimuli. For example, if you present a horizontal bar or a stimulus, then these 3 lateral geniculate nucleus neurons are activated by this bar. Okay. Now, if you change the orientation, now present the bar vertically like this, these 3 neurons are being activated in lateral geniculate nucleus. However, you see that the middle neuron is being activated by both. Same thing here. If you present the stimulus like oblique, like this, the middle one neuron is participating in all 3 situations or stimuli condition. So that's how the same neuron can participate in multiple conditions. Okay. However, these 3 would activate 3 separate V1 simple cells. Okay. This is just a model to show that how fewer lateral geniculate nucleus neurons can send signal to many V1 neurons, because the same lateral geniculate nucleus neurons can participate in different stimulus situation. Okay, so what happens after the primary visual cortex, or V1, which is here, right, in the back, all the way back of the occipital lobe. So here, this is the V1, and V1 then sends signal to V2, then V3, okay, and then goes to V4, and then they go to 2 different pathways. This is the parietal lobe, and this is the temporal lobe. So you see, this pathway is called the parietal pathway, or dorsal, because this is the dorsal of the brain, dorsal pathway. And this one is temporal pathway or ventral pathway. Okay, and ventral pathway goes through the inferior part of the temporal lobe. This is the temporal lobe, so this is the superior part and this is the inferior part. So that's why it is called inferior temporal pathway, and this is the parietal pathway. The dorsal pathway is also known as where or how pathway, because the areas located in this pathway process the signal to determine the location of the object in space, okay, and also motion. However, the inferior temporal pathway or ventral pathway is also known as hot pathway. It helps you to determine the object. What is that? Is it an apple or orange or banana? So hot is processed in the ventral or inferior temporal pathway. We'll talk more about this. So V1 is the early area in the brain, early visual, and V2, V3, V4 intermediate and dorsal and ventral pathways have several areas. Those are the higher visual areas. Okay, now let's see. This is a nice chart. This is very helpful to understand the entire visual pathway from the retina to all the way to the higher areas. So this is really helpful. So let's start from the retina. Retinal ganglion cells. You know there are three types of cells, right? Paracel, midgate, bistratified. And paracel cells send signal to the magnocellular layers of the lateral geniculate nucleus. That means 1 and 2. And midgate cells send signal to the parvocellular layers of the lateral geniculate nucleus, and bistratified cells, ganglion cells, send signal to the kinocellular layers. Make sense? And you also know that through the optic radiation or geniculocalcarein fibers, the signal is taken to the V1 striate cortex, okay? And in V1 you have 6 layers, okay? Only layer 4C and layer 2 and 3, those layers receive input, okay? However, remember, 4C is the main input layer. That means the main layer that receives the input from the lateral geniculate nucleus. And in 4C, so layer 4 has A, B, C, and layer 4C also has alpha and beta. Okay, so 4C alpha and beta, you see, they receive input from the magnocellular and parvocellular layers of the lateral geniculate nucleus, and layer 2 and 3 receive input from kinocellular, okay? Now V1 sends signal to the V2, and V2 has 3 different types of bands, and those bands receive signal from the V1 neurons. What are those 3 bands in V2? Thick bands, thin bands, and pale bands, okay? Thick bands receive input from 4C alpha or A, and thin bands and pale bands receive input from the 4C beta or B, and the thin bands also receive input from layer 2 and 3. Now, thick band has the neurons, those are sensitive to motion sensitive neurons, okay? And thin bands have the neurons, those are sensitive to color, different colors, and pale bands are sensitive to form, okay? So you know that color and form, those are helpful to detect the object, right? Like if I show you a banana, which is yellow and an orange, right? You can see the difference of color that helps you to detect. And if I show you an orange and an apple, both are kind of round, but by looking at the color difference, you can detect the object, right? So what is that? So that helps to detect the object or object recognition, and also form, right? The banana, even if I don't show you the color, by looking at the shape, you can say this is a banana, this is an orange. Makes sense? So the color and form help you to recognize the object or object recognition, and those signals are sent to the V4. So from V2, signal from thin and well bands, that means color and form information, are sent to V4. Okay, and from thick bands, the motion information is sent to the MT, middle temporal area, which is similar to V5 in human. MT is in monkey. Okay, motion, and the motion is helpful to locate the object, and that's why from MT the signal goes to the parietal or dorsal pathway. Makes sense? Pursuing the space and motion, which is where and how, dorsal pathway, and the form and color information from V4 is sent to the inferior temporal that I mentioned before, which is the hot pathway. Okay, object recognition. So you see that V1, early visual, intermediate visual, and higher visual areas. Okay. Now you already know that the V4 neurons are sensitive to color. We know that lack of or loss of color sensitive neurons in the color sensitive cones in the retina causes the color blindness or color deficiency. That is very common, and more common than the loss of the neurons in V4. However, it can happen that your cones are fine. You have all three color sensitive cones in the retina, but your V1 area is damaged, or the neurons are lost in V4. So in that case you will have also color blindness, but this case is much less seen than the loss of cones. However, this can also happen and you will have deficiency in color vision. Okay, that color blindness from brain damage, okay, occurs less frequently than inherited deficiency of cones. Okay, and this is called cortical achromatopsia. A means absent. Okay, achromatopsia. This is something interesting. When we look at something, you know that your eyes are always producing microsaccade. You must remember we talked about the motion or movement of the eyeball, right? Conjugate saccade. We talked about the pursuit, right? And microsaccade. And you know that your eyeballs are always shaking or moving in high frequency, like at least 3 times per second. So that helps to refresh the retinal image. Otherwise, the retinal image will get fade if no refreshment occurs. Okay, and the vision will be blurry. So that is very important, right? So this is something interesting, observation. The subject was allowed to see the face of this girl, and they recorded the movement of or microsecond of the eye of the observer or the subject, and you see the eyes moved frequently. So different area of interest or areas of the face, and by looking at this you can tell where the eyes moved and how much time the eyes spent. So you can see that the observer's eyes spent more time or paid more attention to those particular areas of the face, like here, eyes and lips. So this is a very nice recording that is showing that how the eyeballs move to scan the object when you see something. Okay, now let's talk about the primary visual cortex. So now we know how the signal arrives in the V1 or primary visual cortex, and the primary visual cortex has 6 layers. Layer 4C, alpha and beta, they are the main input layers, receive the signal, and also 2 and 3, right? And from there signal goes to V2, 3, 4 empty, and then dorsal ventral pathways. Okay, and in the primary visual cortex you have the calcarine fissure. That's why the fibers go from the lateral geniculate to the primary visual are called geniculocalcarine fibers or tracts. Okay, and above the calcarine the area is called cuneus. So this is the calcarine fissure, and this part is the cuneus, and below the calcarine fissure this part is called the lingual gyrus. Okay, so cuneate and lingual. Okay, now this is something interesting. If you study the response of the neurons in the V1 or primary visual cortex, if you map very nicely, you will be able to see that there are many hypercolumns like grids, and this is one hypercolumn, this is another hypercolumn, this is another hypercolumn. Okay, so now if you see from the top, you will only record the response. You will be able to tell that they are like this. So this is what you are seeing. Okay, but if you see inside, this is the surface of the cortex. Now if you see inside, you will see they are actually like columns. Okay, and the neurons within each column respond similarly. So these neurons will respond to one type of stimulus. These neurons within each column, all neurons have similar properties. That is the property of the hypercolumn. Okay, so if I insert electrode this way perpendicular and record from this neuron 1, 2, 3, 4, they will show similar response property. If I record from these 4, they will be different than these 4, but these 4 will be similar to each other. Okay, and within each hypercolumn is about 1 millimeter by 1 millimeter grid, and within each hypercolumn you have few things. One is ocular dominance, and also the information of all different directions. That means the direction map, ocular dominance map, as well as the color maps. So all those maps are present within each hypercolumn. So they are kind of independent. They are working independently, each hypercolumn. Okay. Now, neurons of the V1, there are 3 different types of neurons. Okay, input cells, that layer 4C cells, whose receptive fields are the same as that lateral geniculate nucleus and ganglion cells. So this is layer 4C. So this is the neuron of layer 4C, and this is the receptive field. Okay, and this is the neuron of lateral geniculate nucleus. This is the receptive field. And even if you see the ganglion cell, the receptive field will be same as this one, and you can see that on center of surround, right, shown by different colors. Now, when the signal from layer 4C of V1 is given to the simple cell of V1, simple cell has larger receptive field than the earlier lateral layer 4C neurons, right? But still, you see, on center of surround. Make sense? So simple cells of V1. Now, several simple cells of V1 converge on a complex cell. So these are the later cells of the V1. These are the earlier. These are intermediate and late or later cells. And since a number of simple V1 cells converge on a complex cells, you will see the complex cells have large visual field, receptive field, and only they have on center. So those are 3 types of cells in the V1 or primary visual cortex. Here, if you just take a chunk of V1 or only the cortex, you will be able to see these 6 layers in the cortex. 1, 2, 3, 4, 5, 6, and layer 4 is the input layer. 4C has alpha and beta, right? And also 2 and 3. So these are the input layers. 5 and 6 are the measure output. That means they send signal to other areas. Okay, and 4C and 2 and 3 receive input from the lateral geniculate nucleus. Here you see 4C. One thing you can see here, that your right and left eyes input are being kept separated. Even in V1, when they arrive in V1, they are kept separated in lateral geniculate nucleus, you already know, but also in V1 they are still kept separated, shown by 2 different colors, left eye, right eye. Okay. Okay. Another interesting thing happens when the signal arrives in V1. The superior visual field projects to the inferior part of the V1 or cortex. Left projects to right, right projects to the left. Just opposite, flips. Central projects to posterior and peripheral projects to the anterior. You must remember, when the object falls on the retina of the eye, the top goes to the bottom, bottom goes to the top, right? Right goes to the left, and left goes to the right. So on the retina, the object is placed, the image, just in flipped way, right? Opposite. And then when it goes to V1, everything is like flipped again, so goes back to its actual orientation. So this is something interesting, right? And another very interesting thing is that that retina has the fovea, small area in the center, right? And almost half of the photoreceptors are located in the fovea, and another half is located in the outer part, that means outside of the fovea. Although fovea is a very small area, the photoreceptors are present, the cones are present in very high, extremely high density, right? So you have almost half of the photoreceptors in this small area and another half in this large area, right? And when from these neurons the signal is taken to the V1, now I can tell that this is V1, and in the V1, since the V1 neurons are receiving signal from the fovea as well as from the outer part of the retina, the area receives the input from fovea is very big, because from a huge number of cones the area is receiving signal. And you also remember when I talked about three connectivity properties of the ganglion cells, I mentioned that acuity is high in the fovea because each photoreceptor individually innervate each ganglion cell. So from the ganglion cells of the fovea, each V1, right, more V1 receive input than from the peripheral part, because more convergence occur from this part. So that is called the cortical magnification. Although in the retina this area is small, but the area projects from here to V1 is larger in V1. That is the magnification, like called cortical magnification. Smaller fovea is represented in larger area in the V1. Okay, and larger peripheral retina is represented in relatively smaller area in the V1. Okay, so V1 is Broadband area 17, and then association areas are area 18, 19. Those are the areas around the V1. Hypercolumn, as I mentioned, has each hypercolumn. If I just take one from here, this is one hypercolumn. This is another hypercolumn. Okay, so in each hypercolumn you have the no, this is one hypercolumn. So if you just see one hypercolumn, you can see few complete maps. One is the complete orientation map. You see that all different directions or orientations are present in the hypercolumn. That means the neurons are sensitive to systematically sensitive to different orientations. Okay, so you'll find the neurons that are sensitive to different orientations. A complete orientation map. Also ocular dominance. You will see left and right eyes. These neurons are sensitive to left eye, input from left eye. These neurons are sensitive to the right eye. Okay, so these neurons right eye, these neurons left eye. So you see that monocular left and right eyes inputs are kept separated, right? And also you have the binocular neurons. That means the neurons are sensitive to the information coming from both eyes. So you have monocular and binocular, as well as within monocular you have right and left separated. Makes sense? Within the hypercolumn. So that is another thing, ocular dominance. And also you have the color blobs. You have red color blobs, green color blobs, and blue color blobs. So different color sensitive blobs. So all these three things are present in each hypercolumn. So each hypercolumn is working kind of independently. Makes sense? You have the monocular representation, that means left eye and right eye, and you have monocular and binocular representation. This is mono, this is bino, right? They are also present. You have the color blobs, red, green, blue, and you have the orientations, different directions. Okay, map. And each hypercolumn is about 1 millimeter by 1 millimeter area. Two very famous scientists, Hubel and Wiesel, they investigated the V1 specifically a lot, and they found the simple cells and complex cells, the properties of simple and complex cells and their functions. They worked together for many years, and they also work at WashU, medical school, and they won Nobel Prize mainly based on their work at WashU. And the simple cells have smaller receptive field than the complex cells, because the simple cells, like this is one simple cell I showed you before, that simple cells have on center of surround. So this is another simple cell, this is another simple cell. Okay, and simple cells converge on complex cell, and this complex cell has only on center. Okay, and larger receptive field. Okay, simple cells are more useful for edge, stripe, contrast detection, okay, and as well as orientation. However, the complex cell or complex cells are more useful to detect the motion. That means direction and speed. You know, motion has two vectors, right? Direction and speed, right? So the complex cells are more sensitive or helpful for motion, and simple cells are more to detect the edge, orientation, stripe, contrast. Okay, and you see here lateral geniculate nucleus and layers of 4C and V1 simple cells here. Okay, And simple cells of V1 receive input from the lateral geniculate from the 4C input layer neurons, and then here you see simple cells, multiple simple cells converts on a complex cell. That's why the receptive field size is the combination of all these three on center. So much bigger, okay? And here you see how a simple cell helps to detect the orientation more precisely. This is the receptive field of a simple cell, okay? Simple. And you see the middle portion shown by, like, gray or white. This is the on, and outer part is off. Okay, so this is the off, and this part is the on part. Now if you present a bar stimulus, this is the bar that falls precisely on the center, then that will excite the simple cell, right, strongly, because on center is activated. Now if you move that bar to the side, now the bar is on the off surround, right? So what will happen? Inhibition. You don't see any response here. However, you may see some rebound that I talked before, that when you withdraw the inhibition, okay, then some rebound can occur. Okay, now here you see, if you present the bar in slightly tilted or orientation change, like obliquely, here this part is on the on center, so we'll stimulate the on, but this part and this part will inhibit the surround, right? Makes sense? So this part will be stimulated and this part inhibited by that light of the bar coming from the bar. So you won't see, because they will cancel each other, because the stimulation is here, that will inhibit the cell, simple cell, and this part will excite, so they will cancel each other, so you won't see any response. Makes sense? If the stimulus is very big, now if you make the stimulus very large and that falls on both on and off completely, same thing, right? They will cancel each other, so you won't see any response. So you see, only you will see the response of this simple cell when the stimulus or bar is exactly on the on center, okay? And this signal will go to the brain, and brain will be able to tell the location as well as the orientation of this stimulus. If the location, same orientation, but location is here, that is shown here, brain will not be able to determine that orientation. So to determine the location and orientation, you need the simple cells, input from the simple cells to the brain, okay? Now, the complex cell. Complex cells have large receptive field and on center. That means will be stimulated everywhere. If you present the bar here or here or here, same way, the cells will be excited or stimulated. So this is not really helpful to determine the location and as well as orientation, because if I just this is the large on receptive field. If I present the stimulus here, this is the stimulus, the cell will be excited. If I present the stimulus this way in different orientation, it will be similarly excited. If I present here, it will be stimulated same way, right? So same amount of excitation. The brain will not be able to tell the difference of location or the orientation. However, the complex cells are sensitive to motion that I mentioned, right? So some complex cells prefer like particular directions. If I move the bar this way, the neuron will fire. If I move the bar in opposite direction or to a different direction, opposite direction, it will be inhibited. So there are some complex cells, they will like this direction, so they will be excited here, and when you do this, they will be inhibited. Make sense? So the complex cells are not helpful for determining the orientation, but helpful to detect the motion. Same thing here, stripe. This is the picture from a very early study of V1. This is a famous picture published long ago, and you see the V1 and also part of V2 you can see here. And what you see from this picture, that they showed all those three preferences or properties of V1. V1 has hypercolumns and, as I mentioned, ocular dominance, orientation map, and color blobs, color sensitivity, right? So those are processed in the V1. So how they studied, they recorded the activities of the neurons and presented the object in two different orientations, and when they presented the object in vertical orientation, they saw one group of neurons are excited. So certain areas of V1 are excited, okay, other areas are inhibited. When they changed the orientation, like horizontal, and presented the horizontal orientation, the other areas are activated, but previous areas are inhibited. So you have the areas or neurons prefer this, and other areas or neurons prefer this. Okay, so that tells you that you have the orientation maps or preferences in the V1, and they have shown the activation of, you see here, light and dark areas. So light areas prefer one orientation, dark areas prefer another orientation. Okay, so when they switch, one area is activated, and then when they change the orientation, another area again activated. Ocular dominance, they presented the stimulus to the right or left eyes separately. Okay, so when they presented the stimulus to the right eye, left eye was closed, so these columns are activated, neurons in these columns are activated, and when they asked the subject to close the other eye and open, close one eye and open the other eye, and presented the stimulus to the other eye, and then they saw that these dark columns are activated, but light columns are inhibited. So just switched from dark to right to left eyes. So that tells you that you have ocular dominance map. Color maps, when they presented red and green, when they presented the red color, certain blobs were activated. The blobs composed of the cells, color sensitive cells, and when they presented green, other blobs were activated. So you have the blobs for the red and green colors. So this is very nice experiment, although we didn't have a lot of technology to record the activity of many neurons simultaneously, but this was very difficult but elegant experiment at that time. Cortical magnification, I mentioned that the area from the center part of the retina gives signal to a larger area in the V1, and from the outer part, which is bigger, but from the outer part, a relatively smaller area receive the signal. So that is the cortical magnification. Okay. Orientation map. This is a way how the orientation map was determined in V1. So you can insert the electrode through the cortex of V1 and move the electrode like every 500 micrometer or half a millimeter and record the neuronal sensitivity to the orientation. So you first put the electrode here and record the activity of the neurons and present the stimulus at different orientations, change the orientation on the computer screen, and you see that these neurons, like for example vertical, then you move the electrode 500 micrometer and record the neurons response from this area, and you see that these neurons like slightly different orientation. Then you move, you see slightly different orientation. So this way you can determine the orientation map presently in the V1. This is just one neuron, and you see that this neuron, when the electrode was here, like between 500 micrometer and 1 millimeter, this neuron responded the highest to this orientation. This is the orientation, horizontal, preferred by this neuron, right? If you move the electrode a little bit, that neuron will respond to different orientation. So that's how you can create a beautiful orientation map. And this is actually the picture showing how the simple cell of V1 help us to determine the orientation. I explained this. This is the yellow area is showing the on center, and the purple is showing the off surround, right? And yellow is also showing the stimulus. So if you present the stimulus on the on center, then the firing will be very strong. Simple cell V1, right? The firing, because everything is placed just on the on center, nothing is on off surround. However, in this case, you move the bar slightly. Now part of it is stimulating the on center, but part of it is on the off surround. So they will cancel each other. So in this case, you'll see only few action potentials, no activation. Here I already mentioned that 4C alpha and beta, they receive input from the lateral geniculate nucleus, and some input also arrives into layer 2 and 3. So mainly layer 4C alpha and beta, and this is the lateral geniculate nucleus. Okay. This is the last thing you see here, that this is one hypercolumn. Okay, and I actually explained this. If you penetrate the electrode and perpendicular to the surface, you'll see that within each hypercolumn, all neurons prefer same location in space. So by 4 different colors, they have shown the 4 different neurons within this hypercolumn, and you see the receptive field they measure all are highly overlapped. That means they are stimulated when the object is only in this location in space. However, if you insert the electrode like this obliquely, then you can pass through 4 different, here in this case, columns, hypercolumns, and each neuron from each hypercolumn, right? So their properties are different now, and you see the receptive fields are at different locations in this space. So within each hypercolumn, the properties of the neurons are same, and one hypercolumn is different than the other hypercolumn, the properties of the neurons. Here you see that 2 different neurons, A and B, and A prefers this orientation, 90 degree, and B prefers this orientation, 75 degree, right? So we know that different neurons prefer different orientations, which is fine. Now, if you keep the orientation, if you increase the strength of the stimulus here by increasing the contrast of the bars, even if you increase the contrast of these bars, the orientation preference will not change. This neuron A will keep its orientation preference same, 90 degree, and B 75. However, you will see the response of the neuron will be stronger. That means the orientation preference is fixed for each neuron, will not change. However, the strength of response or amount of response could be different, okay, by increasing the contrast. Okay, so let's stop here, and in next lecture I'll talk about the higher visual processing, that means the ventral and dorsal pathways. And in both pathways we have a number of areas, and they process different information. Hot pathway is the ventral, inferotemporal, and dorsal is the parietal, where or how pathway, right? You already know that, so we'll talk about that. Okay, thank you everybody.